Yeah, thank you for the intro, Ricardo, and good afternoon, everyone. And of course, thanks to the organizers for having me on. It's super exciting to be here today and share with you guys some of our recent work, some of which was already published, so that you might be aware of that. And I think it's I will try to sort of explain what some of the things that didn't come into the paper, because that's always more interesting than just going through what you could have read already. And there will also be, because I think it will be also boring just to go through what you already published, there will be some unpublished work as well, which I think will also be quite relevant from the standpoint of going for the large-scale manufacturing, which is, as it seems, the title of the talk. So I think, as the outline indicates, the key aim of the presentation is large-scale peptide manufacturing. And what a better way to start that discussion other than really zooming out and having the helicopter view on things and thinking about the impact of chemistry at scale on sustainability in the broadest possible term. And for that, I will borrow a quotation from this excellent book by this wonderful editorial trio of Tolomelli, Frazano, and Cabri, who has recently edited a book on called Sustainability in Applied Chemistry, which all of you should read, and if you're from industry, you should also definitely buy it. And in the foreword to the book, Professor Paul Anastas, also known as the godfather of green chemistry, states essentially that throughout the history, a lot of brilliant chemical minds have applied themselves to really improve the human condition, only to cause unspeakable harm in the process. And I think this gentleman right here, Thomas Mitchell Jr., chemical engineer and General Motors, is probably the best way to illustrate this phenomenon, because Thomas Mitchell Jr. is most known for two inventions, one of which is freon, and the other one is tetraethyl lead. So that's were pretty consequential advances at this time, but as we already know, they've also caused an immense pain and suffering. Actually, moniker which Thomas Mitchell Jr. has gained over the years is the most harmful, not person, but organism in the history of Earth. So that's not really, that's an achievement in its own right, but not the one which you would like to be known for. So you can ask me, that's whatever harm we've caused as a peptides manufacturing community over the years, it's a bit of a stretch to put us in that category, because that's pretty extreme what the Thomas Mitchell Jr. has done. But if you think about it from the standpoint where we all, presumably we are going from being used to manufacture peptides or oligos, as we've seen yesterday, there's also people gunning for making oligos in ton scale. Let's not forget about that. If we go scale up the chemistry and the purification as is, from the kilograms to the ton scale, all of a sudden we might start seeing what I will refer to as the Mitchell effect on the environment, because the chemistry as is causes some harm at the current scale, but if we scale up things factor 1,000 or whatever the case may be, that will be quite consequential in not a very good way. And the reason why we can't really get away with that is that today we do not have the same level of ignorance as Thomas Mitchell. I don't know what he knew, but nevertheless, what he knew 100 years ago, now we all are perfectly aware of what we're dealing with. So that sort of doesn't really leave us with any choices than really, as we're scaling up, we have to minimize our footprint on the environment simultaneously. So with that, we can move to the peptide, and as we've seen introductions to a number of previous talks, we have quite solid indication of that we're sort of moving to, that peptide therapeutics are kind of moving to the golden age. If you look at the metrics like a number of launched peptides in the clinics, or the projected market caps, all are trending in the direction of quite significant growth. Not least also thinking about almost intoxicating success of some of the recent incretin analogs. People are anticipating that this trend will continue, but it has also generated some downfalls. Notably, from the standpoint of capacity. Some industry veterans are like, decades ago we were celebrating opening champagnes every time there was a kilogram or 10 kilograms batches of peptide manufacturing. Now we're talking about ton-scale peptide manufacturing as a regular occurrence, really? So that's one kind of a question mark. The other one is, of course, if you look at the amount of the waste, hazardous waste that we produce, and you scale it up factor 1,000, you do the math on that as well. So there was actually a recent roundtable interview in the Chemical Engineering News where the forecast was that of course, the emphasis on quality will not disappear, but the increase in the volumes and demand of more sustainable manufacturing will go in hand in hand coming years. So how do we try to address that at PolyPeptide? I will really not dwell too much on that because it's something we can, the information you can find on our corporate website is that we are committed to sustainable practices as the CDMO operating in the tide space. We, of course, have a range of initiatives aimed at increasing the capacity, but it's all anchored in the, of course, keeping the quality and sustainability, and we have no biases, no ideologies when it comes to technology, whether that's with internal or external collaboration. We look at everything, turn every stone, and that doesn't mean we implement everything, of course. So we implement what we really think makes sense on the bottom line. So what I will spend a minute on explaining, though, is how, because that's a question of everybody's lips, is so how are you going to make a ton of peptide exactly in a year? So we kind of coined this phrase aimed at that feature called modular approach to large-scale manufacturing. And just to break it down for you, we kind of think that the way forward is to use standardized both upstream, downstream equipment based in modules, sort of like a Lego, if you will, and then you can use these modules in a flexible manner across the globe, wherever you have your manufacturing facility. So I will illustrate that by a manufacturer of a LIS branched incretin analog. You can sort of find the details of this process in the reference in the bottom of the slide. It's not exotic from the standpoint of the overall flow of process flow. You already start with a relatively high scale SPPS, 13 mole. 600 liters reactor, that's one module, then of course the cleavage, DSP layout could be 2 modules, but there is one, gives you 15 kilograms of the API. Well, that's quite far away from making a ton, you will say that, and that's fair comment. But actually, the projected output for this one line per year, and that's real data, so it's legit, it's not like pipe dream by any stretch of the imagination, it's actually 700 kilograms. So how do you do that without like having 20 of these reactors, or having a reactor which is 10 times larger? That would be very, very good from the risk mitigation perspective. So what you do basically is you optimize your SPPS to the extreme, most notably from the standpoint of minimizing critical impurities, because these have the most profound impact on your downstream processing, where most of the loss of sustainability can actually happen. On a key aspect, I don't think you can see it on this image, but the reactor has to be temperature controlled, because the chemistry which you can carry out at room temperature will be by definition suboptimal. So if you can work with the temperature, which we can even at this scale, you can optimize the things to much higher extent. So you can do coupling cycles of less than 4 hours, so even if you do some life cycle assessment of 3 hours, and then you can do all the races, then you can do your 1 SPPS in a week together with, of course, aligned cleavage and DSP. Now the equipment is dedicated, so you don't have to do cleaning, validation, and all the sort of things, so you can kind of crank out about 50 batches a year, so that way you arrive at the 700 kilos API a year. So if you need more than that, of course, you can set up more production lines, and that way you will get where you need to be from the standpoint of capacity. So that's an approach. Whether it's the best one, you can discuss, but it's certainly viable. So with that, we can move to what was by the previous speaker referred to as the elephant in the room, and it is. Now I was showing you that we were kind of betting on this SPPS as being the key element of sustainable peptide manufacturing. Everybody will tell you, well, SPPS, is it really sustainable? Well, to that I will say to render, so we actually wrote in this book edited by Tolomelli and Franzano and Cabri, we wrote a book about this, book chapter about this sustainable SPPS, where we kind of preach the idea of an integrated approach to making SPPS truly sustainable. Yes, a lot of people are looking at the amino acid cycle. You replace the DMF and what have you. You can also replace the DMF in the first amino acid attachment. To render the SPPS truly sustainable, you have to play this movie in your mind all the time, where you also worry about what's happening with the starting materials, the cleavage, how do you make even the most complex peptide, how do you minimize the waste. Correct? No problem. So this is something you have to consider concurrently. This is not something, you cannot have this like approach where you just have your view set and just let's replace the DMF with the green solvent and you're home free because you're not. So the conclusion of the chapter is that it's reviewing all our advances as well as everyone else working in the field. So I encourage you to read that, but it's kind of an open-ended chapter because the notion that there is that it can be expected, but there's so many people working in the field that the momentum is really gaining some traction that eventually this SPPS will rise as a phoenix and really fly high with not being just super flexible, versatile, and now I showed you scalable, but it will also be a highly sustainable peptide synthesis platform. I think it would be a stretch to claim that today, but I'm personally convinced that that's where we headed, and we have some true giants in the field sitting in this room, so we can discuss how we'll come to that end. But with that, I will just give you some case stories. The rest of the talk towards the theme you saw on the previous slide, because as I just mentioned, we're going for growth without trying not to compromise the quality of sustainability. And in doing that, you have to acknowledge you cannot just carry on as you've already done. Some of the well-established practices or materials you've been using, you have to rethink quite considerably, if you will. So the two things I will touch upon today is peptide resin cleavages. Of course, TFA is another big elephant in the room there. And I will also touch upon the role of quality of starting materials as it pertains to the sustainability of large-scale peptide manufacturing. At the end of the talk, as a bonus, I will actually show you how these two themes influence each other. That's unpublished data, so you can hopefully learn something new today on that. So we're starting with the peptide resin cleavages, just a bit of a quick historical perspective. Merrifield's SPPS, from the standpoint of cleavage, was actually quite sustainable because, saponification with aqueous ethanol, not too shabby from a sustainability standpoint, that's fair enough. But very quickly, he got better ideas. He invented box SPPS just a few years later, which was from the SPPS standpoint was a vast improvement, but from the cleavage standpoint, not too great. Initially, he used a two-step cleavage because he didn't know how to cleave the peptide of the resin and side chain group at the same time, so he used hydrogenalysis for that. But he wisened up. At that time, everybody thought he used HF. Of course, we now know that's not really an option anymore. And even the alternatives for HF, which you see in the literature, are not that great. But that's actually not that much of a problem because I wouldn't say that the box SPPS is wiped out from the face of the earth, but it doesn't really play a meaningful role in the manufacturing not going forward. That's not really that's not what we see nowadays. So of course we all know that what the ultimate peptide synthesis and scale-up manufacturing workhorse is FMOC SPPS, and that from a cleavage perspective, of course, is let's be honest, we've all been the slaves of TFA for many, many decades. And, that's the devil it performs extremely well. It's universal, and it's we can do a lot of beautiful things with it, but it's not sustainable. It's extremely harmful, and it's also the so-called PFAS chemical. And so there's been some indication of that could be reversed. Like 10 years ago, more than 10 years ago, Stechenko and Palladino came up with 2 papers where they introduced a mixture of 2 Bronsted acids, hexafluoroisopropanol with aqueous HCl, and showed some preliminary data that could be a direction to go with. However, that bird hasn't really flown since. If you look at the paper, the follow-up paper in which they looked at the scope, they got out with 2 5-mers where they could cleave like an ester and ether, and that was it. So that's not really that great. So basically, we are in the area of total dominance of TFA, which is not that great from the sustainability standpoint. Of course, that's how operators using TFA are supposed to look at the polypeptides. It's not really the ideal outfit, if you ask me, but that's what it takes. So the pressure to eliminate PFAS is certainly increasing. There are some harsh words from the regulators on both sides of the pond. And yeah, that's not really an indication of that would be, the pressure would be lessening anytime soon. So if you look at, for example, in medicinal chemistry, this molecule, this anti-HIV, a small molecule has made the news recently, not because it is efficient in phase 3 clinic, which it is, by the way, but it was questioned as being PFAS, 10 fluorines per such a small molecule. Now even medicinal chemists are asked to get rid of the fluorine, and fluorine used to be the magic element for many decades. So that's, that will mean something if that is not going to be the avenue you would be able to use. And even in areas as far remote from the medicinal chemistry as sports, this skier here was banned from, was disqualified from a race for having detected the fluorinated substance on her skis. So that kind of goes on to show you that the societal pressure is increasing, the writing is in the wall, and PFAS-free alternative for TFA is urgently needed. So if you should scrutinize how it should look in practice, you have to look at the actual workflow of the peptide resin cleavage in the context of FMAC SPPS. Of course, you have TFA in the cleavage. That's really a problem. Then you have the precipitation, which oftentimes happens as hazardous anti-solvent. And then you have unit operation like centrifugation or filtration, which is not that great from sustainability standpoint either. So you should do some things to improve sustainability. You could do, first and foremost, of course, improve chemoselectivity if you can, because that improves vastly sustainability downstream. But you should, of course, look at use less hazardous materials and practices throughout the entire workflow. And you should also look at that not just for the global cleavage, but also for the so-called soft cleavages, because you use TFA there as well, or more specifically TFA DCM, where you can cleave the fully protected peptide fragments, or you can use TFA DCM to cleave side chain protecting groups and do some things from there. So none of which is that great from sustainability standpoint, so you could also do something about that. So before we get to TFA-free cleavages, just a few quick words about some of our earlier work in the area of TFA-based cleavages. How do we explain the sustainability in that? You already seen the previous presentation that we some years ago came up with BDMT as being conceivably an improved scavenger for the conventional aliphatic scavengers. I cannot really do a better commercial for that than the previous speaker, so I'll just leave it at that, and you should try it on if you have some cumbersome cleavages, like very correctly we've also seen with the pseudoprolines. These can be kind of quite sticky, not willing to deprotect, and the advantage of BDMT it has, that's just a theory I have, but it can be a different explanation. It's actually much more stable in the TFA than DTT is. So if the DTT kind of dies too quickly, then it cannot deprotect your pseudoproline. You will see it in the paper, the data, that the half-life for BDMT is appreciably higher than for the aliphatic thiols. So another things we did with green cleavages was TFA-free side chain protection. We didn't skip the TFA, but we were able to replace DCM with this acetonitrile ethyl acetate mixture. Of course, deprotect the PIP and the MTT on the aspartic acid lysine. And then you, of course, can cyclize and do that sort of thing. Another thing you can do is fragment coupling with the fragment cleavage with the low concentration of TFA in a non-DCM solvent, which we also did, and that's that same paper referred to here. Here it should be noted that the Latorre and Albricia group and Al-Musaimin groups have taken this concept to the next level and showed that replacing this solvent and some other ones you can actually gain considerably in the scope. So the soft cleavages without DCM are now quite feasible if that's the direction you want to go in. So moving on to the TFA-free cleavages, there's some key consideration. I mean, this is something you really should think through before you get going, because if you don't think very hard, you might end up with the result which will not really work for you that well. Because what you need to do is really have something which replaces TFA, but you don't want something less chemically suitable. That would not really be. That would be like replacing the fluorinated wax for the skis with non-fluorinated. Everybody knows that just gives you a worse skiing experience. So you do that because you have to. But ideally, you would replace it with something which is not PFAS but works just as well. And of course, time is of an essence. With the increasing pressure of the PFAS, you don't have like a 5 to 10 years horizon. You have to move very quickly. So you could go for acid-free conditions, scale photocleavable linkers and such, but that would most likely require that you would revise your, the classical FMOC SPPS scheme. Not so sure that the conventional users would be willing to do that. They would like to stick to their guns, their preferred amino acid, preferred linkers, so on and so forth. So you could try some bronchic acid or bronchic acid mixtures. It's Tsetsenko Palladino did, but that tends to work okay. We did quite a bit of work on that for simple substrates. Once you go to the very complicated substrate, you might end up getting in trouble. So we kind of thought we would set out with trying with loose acids, and you can see that the loose acid have been tried for some cleavages of linkers and isolated instances of protecting group. Again, this subject has been reviewed recently in this book on sustainability in time, so you can read all about it there. So that could be a starting point, if you will, and we also wanted to, as I alluded to a couple of slides ago, sort of see if we can skip the unit operation of precipitation and just do the dilution and just off to the races to the DSP, because that would be much more sustainable as well. So our initial results were not that great. You can see that this FMOC HMPS MS acid, this is the cleavage with TFA. And iron trichloride with the Mandar group, iron trichloride and DCM with the Mandar group was able to show in solution and also in resin that removes the Lisbok and aspartate quite efficiently. We were not getting nowhere with that. Of course, we tried to go on to the acetonitrile because that we could that's more sustainable DCM, and then we could just dilute our cleavages with water, and that would be, of course, very convenient. Nothing was happening, not even with any other loose acid. So after some experimentations, we actually came through with this system, just using acetic acid, acetonitrile, a little bit of ferric chloride, and the heat. Voila, you have a full conversion. And I will just let stand there for a while for the ferric, because ferric chloride, acetic acid, acetonitrile, not exactly the three most exotic chemicals you will find in the closet. So the fact that somebody hasn't done that like decades ago, that I had to stumble upon it like relatively recently, kind of tells you something about it, that we need to be more radical in departing from our preferred approaches. So with that, we kind of moved on to a lot of Bronsted acid, loose acid solvents. I will not go through the details. You can find it in the supporting information to the paper. So we move on to the more complex peptide with the Bronsted acid, loose acid solvent system that we deem suitable. And the question is, what kind of peptide resin you want to be tested? Of course, you want to see the test all the common peptide protecting groups and the linker types. But one feature particularly we're interested in testing is to test peptide resin, which were prepared by green SPPS, and the reasons for that, and this is the recent review we did on the green SPPS method in this chapter 4 in the Sustainability in Tides book, is that you see that starting with the Albricious acetonitrile and using PEC resin back in 2009 to this dipropylene dimethyl ether by the Sala group last year. There's, of course, some more paper still coming, but there's a common denominator, and that is, of course, that all these green methods were utilizing TFA. And if you ask me, if you use DMF, if you free SPPS process and TFA in the cleavage, you cannot really deem that as truly sustainable. It'd be a little bit like if you had a headache and the cancer and you took some headache pills. You wouldn't have a headache. Well, guess what? The cancer is still there. And in this case, the cancer is certainly a TFA. So, and if it seems to you that I'm pointing fingers, well, certainly I am, and more than anybody else, I'm pointing fingers myself, but because I had the responsibility for the fair share of the papers in this table. So going forward, I think the mission is clear that the sustainable SPPS has to be both DMF and TFA-free. So with that, we chosen a resin which we prepared some years ago by process using ethyl acetate DMSO, as you could see in the previous talk. That has become implemented quite regularly nowadays, and we actually also find out that these solvents can be recycled, so you can minimize PMI quite significantly. So we kind of want to make that coupling between the green SPPS and TFA-free process, and we started with as I told you, we want to also get rid of the precipitation. So we kind of looked what kind of impact the diluent has after cleavage. You can see that if you dilute with acetonitrile water or acidic water, you have a whole. It's kind of amazing what kind of different breakdown patterns you can see in the post-cleavage dilution. So, but if you use this ammonium acetate, it's kind of a useful trick because that your peptide is diluted to a pH 4, and that's usually quite suitable for most peptides. So we kind of use that throughout. And for this AIB ACP peptide, which has this amide, ester, ether, or two amides, a Ramage linker. So after some experimentation with the temperature and the ratio between the two, between the loose acid and the Bronsted acid, we figure out that we, with slight heat and approximately 1-to-1 ratio of the between the loose acid and Bronsted acid, now you have suitable protocol for these cleavages of this peptide resin prepared by green SPPS for both ferric chloride, acetic acid, ferric chloride, HCl. So that was quite encouraging, and we then tried another 10-mer peptide, a pharmaceutical relevant peptide is anti-prostate cancer degarelix peptide resin, which the biggest reason was actually to go from Ramage, which is kind of very cleavable by a low content TFA. You don't want people to call you out for that, so we took a ring amide, which is of course much more tedious to cleave, and we've shown that again what was important there was again the ratio between the HCl and ferric chloride. If you have only ferric chloride, nothing happens. 1-to-1 ratio, you get a very pure product, but not very high yield. But you can see that the addition of the extra HCl is not really improving things. That causes side reaction. Again, you can read all of these 23 experiments in the paper. So we wrap things up with just looking at all conventional amino acid PGs in the which you would encounter proteinogenic acid. These are, of course, to be expected because they were already covered. These were not covered in the two substrates you saw on previous slides, and you can see from the number of experiments, some of them were kind of pulling my teeth just throughout to get those to work. So you can see all the specifics of that, but eventually in the paper and its supporting information, but eventually for each and every one of them, we were able to come with protocols that are taking care of those. So I will just show you one example of cleavage of one specific combination of amino acid protecting group, f-methoxystridyl. And the reason for that is it was quite unique what we encountered there. First experiment was not that good at all. If you can see TFA, DCM, you get a very clean f-methoxy product. No product with ferric chloride, HCl. However, a slight amount of engineering, you go from HCl to acetic acid, and add the DTT as a scavenger. Now you have the reduced FMOC-cis in a very good purity. And the story doesn't really end there, because if you remove the DTT, now you have your substrate in the oxidized form. So that doesn't really take much of an imagination to sort of realize that kind of opens up for way you can work with this. by design manner that you can make heterodimers, homodimers, cyclized peptides, what have you. So that's something we'll be looking for in the future studies. And of course, a fair question is to say what happens with the iron. So we kind of showed. This is of course also important to say that the crude from TFA cleavage and Bronsted-Lewis cleavage essentially undistinguishable, and then you just remove the iron by just a simple SPE down to 80 ppm, which is well below what the authorities would expect. So, just to summarize the TFA-free cleavage work, first the DMF and TFA-free process, it's not really a paradigm change at this scale, but that's certainly what we're gunning for. And of course, the dimethyl carbonate and acetonitrile open up for being able to dilute rather than precipitate. These are some very simple chemicals, so that really, there's no real some warning sign that we wouldn't be able to use it in scale, and that's exactly what we're gunning for. I didn't tell you that until now, but we used HCl in dioxane, which is normally what you do in a small scale, because that's what's the most readily available form of HCl in R&D, but we, of course, working very hard to replace it with greener media. And of course, another point on the near agenda is to demonstrate that this will work suitably well in, let's say, 1 kilogram, 10 kilogram resin cleavages, because that's what you normally would do on an industrial scale. So with that, we move to the other theme, which is the quality of starting materials. It's a very important aspect in pharma manufacturing. Just highlight two of them. In the recent literature, all of you might have heard about HCl and acetonitrile, HCl formed from coupling reagents, how to prevent that. But you can also have HCl and acetonitrile. You can also, another problem, you can have formation which can also wreak havoc on the downstream processes. So you can also form formaldehyde if you have a residual DCM in amines, forming this adduct, which then breaks down and hydrolyzes to formaldehyde. So that's something you should be aware of. So in a peptide manufacturing, you would typically have a risk-based approach to control of the critical process parameters, which they will then secure that you control your critical quality attributes of your API. And of course, from the standpoint of starting materials, you would have controlled the critical material attributes, whether it's amino acid. Of course, everybody knows you have to be very concerned about quality of those, but you also have to be concerned about the quality of all the other things, as I will show you. And as you go into more sustainable processes, remove DMF and replace it with other things and so on and so forth, you should never compromise the quality. And of course, to be able to control the SMs, you should really need to understand the impact of the attributes on the process, and you need the suitable analytics as well. And we did some prior work on that. I will not really go through that very quickly. You can just read about this in this series of papers we published on the subject. There's the DIC, you might be familiar with that reagent. That's probably the most common reagent in SPPS. We discovered some years ago that it contains this thiourea, which prevents radical-induced side reaction. Then when we studied this quality of the side, this reagent some years later, we actually figure out that now it contains mostly this diazetidinone, which is a product of the reaction between DIC and isocyanate, which is not a radical scavenger. So your own quality of that starting material variation can really influence whether you see oxidants or not. And we were later on where the Latour Alberici group introduced this, let's say, improved version of DIC from many aspects. Now it's getting implemented by more and more groups. We also looked at the quality of this reagent and found that similar varying amounts of similar impurities are also present in this one, so you should take that into account as well. We also looked at the quality of the solvents in the recent study, showing that this green solvent called NBP contains and can also form during standing and chemistry this hydroperoxide. It can also do damage to your process, and which ironically, we kind of could leverage the finding about DIT has actually turned out as a good suppressant and those side reactions as well. So it's kind of become a little bit of a Swiss Army knife for us from that respect, because regardless of whether you have a problem in the radical-induced side reactions on the ones induced by the hydroperoxide, you can just check in a small amount of DIT use. So things turn tend to work out quite well. So I'm not discouraging anybody. That's a kind of a disclaimer. I'm not discouraging. I'm not here to discourage people from using NBP or any of these reagents. On the contrary, I'm just saying you should do your due diligence and really think how the potential quality of impurity presence can influence your process. And you really need more adequate analytical means because currently everything is about GC and GCMS, and the compounds like DIDT or the NBPA hydroperoxide are not stable in these methods. You need LCMS to be able to assess them. So that's something you should ask your vendor to consider. So with that, we actually move to the last part of the talk, and it's assessment of the quality of SPPS resin, which we embarked on quite recently. And you can see these are a bit more tricky to analyze than things like amino acids, reagents, solvents, and what have you, because these are not that easy to analyze by LCMS unless you really dissolve them in some mixtures of bronchic acid and stuff like that. And you can't really use them in SPPS that well. So you need some mixture of direct, indirect methods. Sometimes you use this. Oftentimes we're not very satisfied with the quality of the SPPS resin, and I will just talk a little bit about quality of the Wang resin. And the reason for that together with CTC resins, these are the two primary workhorses for the synthesis of peptide acids. Many people have tried and have died on the hill of trying to replace those two resins. They're still out there. PolyPeptide, we use Wang resin to the tune of 100 kilograms a year. So the question, we have seen some, upon changing vendors, getting new batches of the resin, there were some batch-to-batch variation. Could that be linked to the quality of the Wang resin? How do you determine that? So you probably should start by looking at the route of synthesis of Wang resin. That is the classical way to make Wang. Of course, polymerization of DVB with styrene. Then you do the Friedel-Crafts chloromaturation, as the process is called, and you, of course, take the 4-hydroxy benzyl alcohol on a base, gives you a Wang. Whole bunch of side reactions, difficult to control ones, can occur both during the chloromethylation and also esterification. So the question is, as we are also dealing with Wang resins from alternative vendors, which were using the so-called copolymerization process, now you use the DVB, styrene and chloromethyl styrene to get the Merrifield one part, and then you use the same step in the esterification to get the Wang. So will we be able to see some differences in quality between the two route of synthesis to Wang resin? And so we actually studied this very thoroughly, very complex testing regimen. I'll just show you a snippet of that work for 4 of the batches which we tested. 2 were made by the copolymerization approach, the CP, and 2 were made by the Friedel-Crafts approach. And so what I will just zoom in on is the impact on the quality of the Wang resin on this cleavage process. And that's depicted here. So what we basically have done, we loaded the Wang resin with ethmoglycine, gives you ethmogly Wang resins. And when you cleave that, notice in particular the impurity at RT 11.4 minutes, and that's significantly higher for the 2 batches of the Wang resin prepared by the Friedel-Crafts approach compared to the copolymerization approach. And you can see that's not something which was the ethmoglycine which we loaded onto the Wang resin. Could that be something that we did something funny during the loading that has influenced this variation? Well, we wanted to test that. So what we did was actually TFA cleaved the naked Wang, if you can call it that, without any amino acid attached whatsoever, and I could still see this RT 11.4 minute impurity. It wasn't there in the blank. You can see that this little peak that just comes from blank TFA, and we tried to DMF cleave the Wang resin. Nothing came off. So whatever came off, and it comes off in a higher amount with the Friedel-Crafts Wang than it did for the copolymerization Wang, was TFA cleaved. So that was quite interesting, because that would indicate that if you had at your hands a Wang made by the polymerization approach, you could anticipate lower content of impurities as compared to the Wang made by the conventional Friedel-Crafts Wang. So that was quite interesting, and we were kind of course, wondering what exactly is this impurity. And to that end, we did some test cleavages. This is, of course, the cleavages of the naked Wang. Gives you the RT 11.4 minute impurity. you would think that it was related to the 4-hydroxybenzyl alcohol linker, and you can see it's not the linkers itself because the linker comes here, but if you mix TFA and 4-hydroxybenzyl alcohol, you arrive with a mixture of compounds, one of which, and we confirmed it with the mass spec, is actually the 11.4 minute impurity. So that demonstrates that whatever comes off the Wang resin, it's actually related to the reaction between the TFA and the 4-hydroxybenzyl alcohol. Most likely, of course, sans evidence, that's what I have to say, is that the so-called 4-hydroxybenzyl cation is involved. And that cation is actually very well known to wreak havoc on the electrophilic residues like tryptophan, because then it will form this plus 106 Dalton adducts, and you can encounter that with the ring amide linkers as well. So that's something you can anticipate, that if you go from the Friedel-Crafts Wang to the copolymerization Wang, you should be able to get lower content of this plus 106 Dalton impurity. So that's pretty valuable information in its own right, but that's actually not where the story ends, because as I was preaching to you at the beginning of the talk, we should all get rid of TFA. So now I'm taking you through some slides, but let's use TFA for cleavage of Wang. That's of course untenable. So what happens if you look at try the TFA-free cleavage see how it works versus the quality of the Wang resin. So what we could see is that if you take the Wang resin, cleave it with TFA DCM, as we could expect it, we get this TFA plus 4-hydroxybenzyl cation impurity. When we use the conditions, which I showed you a couple of slides ago, which cleave F-MOIC amino acids off the Wang quite well, ferrichloride, acetic acid, and acetonide, it was in heat and it was for a long time, nothing comes off. So what I'm actually telling you is that I would like you to consider to go away from TFA and try something else, such as these mixtures, bronzite acid and Lewis acid, not for the sake of getting rid of just of the TFA as being hazardous, being PFAS. It actually also, and we're looking at it as currently, we will potentially address a quite painful chemoselectivity problem. So that's probably a commonsensical reason to go from one cleavage cocktail to another, if you will. So with that, I will just summarize that, as I showed you at the beginning, there is the tidal shift to very large-scale manufacturing in a sustainable manner, both for peptide therapeutics is well underway. We're not there yet, but I think the outlook of that endeavor is very bright, and I think that I hope I was able to outline for you, since the large-scale data, that TFA-free cleavages and paying almost autistic attention to if you will, the quality of your starting materials. Yeah, it's a bit of a weird term, but it's true. You have to be really, you really have to. When it comes to quality of starting materials, devil is indeed in the details. So you should really be concerned with the quality of your starting materials, other than the F-MOIC amino acids. And last but not least, I started with a quote from Anastat from Forward to the Sustainably in Tides book. I've also finish off with it. Of course, we should always aim to reduce, recycle, and reuse. But other than that, we will need radical and practical innovations on top of that to get where we need to be from the standpoint of sustainability in peptide manufacturing. And with that, I would just like to thank some of the many people who were involved in the chemistry show during this presentation, most notably Christophe André, who was my key player and partner in crime in the TFA-free cleavages work. Sarvat and G were involved in some of the work on the quality of starting materials. So all of these talented analytical chemists were doing some of the key work to be able to analyze all these products of cleavages and also the starting materials in an appropriate way. And of course, you all of Luxembourg has been supporting us, not just us, but presumably many others. And he really is a key player in this industry. Some people might not realize it, but he really needs to be thanked a lot. And yeah, and with that, I would also like to thank you for your kind attention. I'll be happy to take any question now, or yeah, whenever. Thank you.